right, welcome back for part 2 of the innate immune system, chapter 16. In this section, we're going to talk about the different types of white blood cells. Some of those are called granulocytes, some of them are called agranulocytes. We're also going to differentiate between lymphocytes from the other white blood cells. White blood cells are leukocytes, and some leukocytes are lymphocytes. So we'll talk about that. And we'll talk about the amount of each white blood cell normally in the blood and the effects of infection on those numbers. So using a differential white blood cell count to see if a patient has more or less white blood cells than normal. So blood sample, blood test can be taken and centrifuged down. So when you place blood in a tube at first, it's all mixed evenly. It would all look red, right? Then you put it in the centrifuge and it makes these layers. And the top layer would be clear to yellow. And this would be the plasma portion. This contains mostly water, but it has ions, proteins, all the nutrients that's going right from the digestive system that's entered into the blood to go feed all the cells of the body. It's also going to have lots of waste in it because as the blood circulates through the body, it picks up waste, right, from cells, from that interstitial fluid. And then gases is that's part of it, right? So bringing in oxygen and taking out CO2. So that's plasma, lots and lots of things going on with the plasma. Then in the middle, you'd have a thin layer called the buffy coat, and this is where you find the leukocytes. Remember, white blood cells are called leukocytes. We also have our platelets, which you know is for part of what is needed for clotting. And then below that, you'll have the layer of your red blood cells, erythrocytes, okay? And the process of making both red and white blood cells, so all of these red blood cells, white blood cells, and also the cells that make platelets, all that comes from your red bone marrow. You have stem cells in your red bone marrow that produce all the cells of the blood, right? And so that's process of making those, the process of the stem cells making all the different blood cells is called hematopoiesis. And there are a few different ways you can organize the blood cells. This is showing the lineage of which cell then produces which cells. So the very first differentiation step from your pluripotent stem cells in your red bone marrow, you're going to get two types of cells, a myeloid progenitor and a common lymphoid progenitor. And the myeloid makes your cells that lead to your platelets. It makes your red blood cells. It makes mast cells, which recall are cells which are throughout the body, but especially concentrated in your skin. And they have these granules to help if those cells become damaged, they will then release granules that will release things like histamine to initiate an immune response. We will only mention mast cells maybe once or twice, as our focus will be on other cells. But I did want to make sure to bring that up. You can also read about mast cells or refresh your memory in your textbook. Okay, and then the last type of cell made from your myeloid progenitor is the myeloblast, which makes white blood cells called basophils, neutrophils, eosinophils, and monocytes. Okay, so the myeloid progenitor will make the myeloblast, which will then make a majority of your white blood cells, basophil, neutrophil, eosinophil, and monocyte. Monocytes circulate in the blood. When they move into the tissue, then they will further differentiate or mature, and they will either become a macrophage or a dendritic cell. Okay. Now, the lymphoid progenitor makes your lymphocytes. So that's a nice name, right? Lymphoid makes your lymphocytes. And the lymphocytes are going to be either T cell or a B cell. But also, and this is where it can get confusing, is that the natural killer cell is also made from this lymphoid progenitor. But the natural killer cell would not be considered a lymphocyte. It also would not be considered. So these T and B cells are what we call agranular. And the natural killer cell, as you see depicted in this image, have large granules. So that's why I've mentioned there are many different ways that you can organize these cells. This flow, this organization that you're seeing here is only the lineage. So I added a slide so you could have that written down for sure. So make sure you write down that this flow is only showing you the lineage of which cell produces which cell, okay? This is not showing you how they stain. It's not showing you how they function, right? It's just the lineage. All right. Staining, on the other hand, will give us the two groups called granulocytes and agranulocytes. The granulocytes are the cells that you can see large granules after simple staining and viewing under a simple microscope. We can see neutrophils, basophils, and eosinophils very easily. So neutrophils, you can use both a combination of acidic and basic dyes, and that will stain the neutrophil a pale, pale purple, pale lilac color. And then what you'll see, in addition to all those granules, you'll see a funny looking nucleus, which is called a polymorphonuclear leukocyte, because it has these 2 to 5 lobes, and you have I can actually show you in this image. So see this neutrophil has 1, 2, 3, 4 lobes in this image. Sometimes you can see 5 lobes. Sometimes you only see a couple. But this is polymorphic because it can take on different shapes, different numbers of lobes. And before looking at these white blood cells, you're used to just seeing a single one round nucleus. So now we're seeing a very different type of shape. So the neutrophils are the ones that have several lobes, okay? But they're called granulocytes because of the large granules. So it's all the dots everywhere throughout that cell. And you have another image in your textbook that you can look at as well. Basophils, you'll use a methylene blue dye, which is a type of alkaline or basic dye. It's also a very simple dye, very common. It'll stain it purple blue or blue purple, and you'll see lots of large granules, as you see in this image here. And again, you can look in your textbook to see it up more closely. And then eosinophils, which you can use eosin, an acidic dye called eosin, and that will stain it like a red-orange color. And so you see here, this cell is like a reddish color, and you can see lots of granules. Eosinophils also have an interesting nucleus. They have this 2-lobe nucleus, okay? And then that's pretty much it as far as what you can see under the simple light microscope with simple stains. for granules. Now, the A granulocytes will be your monocytes and your lymphocytes. Although you can cross out natural killer cell, because natural killer cell, as noted here, does have large granules. And so we will just ignore or cross out the natural killer. So here, let's go ahead and do that. Pen, magenta, cross out. Okay. Now we'll go back to my arrow. Okay. So staining again is using a regular light microscope, and the leukocytes can be broken into the two groups based on their staining, granule and agranule. But that the names are a little misleading. So it's seeing these large granules under the microscope, but the agranules do actually have lots of granules in their cytoplasm, but the granules are small enough to where you don't see them under the microscope. Okay, so that's what this is about, reminding you that when we say granulocytes and agranulocytes, we just mean under regular light microscopy. All right, now the more important way to look at these cells is their function, right? So neutrophils are the most numerous white blood cell in circulation. They are highly phagocytic, and we will talk about the steps of phagocytosis in a little bit. They're motile. They are able to move through from the blood into tissue and move throughout the tissue. They're most active in the initial stages of an infection. Okay. And as I already mentioned, they can leave the blood and enter infected tissue. Eosinophils are mostly used, or they're most active when there's an extracellular parasite, which is too big to be eaten by a phagocyte. So eosinophils will attach to the surface of the parasite and release or throw all these granules at it, including peroxide ions, to try to kill it that way. The eosinophils are also possibly related to allergies as well. Basophils are more related to allergies because they're specifically releasing histamine. And then monocytes are, as we talked about, they're these like immature phagocytes that circulate in the blood. So in the blood, the monocytes have no activity. They're not phagocytic in the blood. They have to be attracted to the site of an infection, enter the tissue, and then they become activated. And they can become activated into a macrophage, or they can be turned into a dendritic cell. Macrophages are, we'll be talking about them a lot. They are mostly seen in like the later stage of infection. They're good at phagocytosis, but they can also cause a lot of tissue irritation. And so like I said, we'll talk about macrophages a little bit more later on. Dendritic cells may also come from monocytes, and they have those funny looking extensions that make them look kind of like neurons, and that's where they get their name dendritic, because they kind of resemble dendrites, right? They're phagocytic, and in addition to engulfing a pathogen, they can also then, what we say, process the antigen from the pathogen and display it on their MHC self-tag, which helps other immune cells, T and B cells from the adaptive immune system, to know that there's been an invader. And so it plays a very important role in the activation of the adaptive immune system. There are other what we call APCs, antigen presenting cells, But dendritic cells are especially important group. Okay, abundant in the epidermis, mucous membranes, and lymph nodes. So dendritic cells will be found in the highest concentration outside of the blood, right? In the skin, mucous membranes, lymph nodes. Okay, now natural killer cells are their own group, and they are part of the innate immune system. They will look for cells that have been infected or cancerous, precancerous. These are cells that are no longer behaving correctly, and they will lose their MHC self-tag, and then they will be killed by natural killer cells. And so I have a little slide about natural killer cells just to clarify what natural killer cells are, because students tend to confuse natural killer cells with cytotoxic T cells. Cytotoxic T cells are part of the adaptive immune system, and they function in a different way. Okay, so natural killer cells non-specifically destroy infected cells. This could be a bacterial infection if it's an intracellular bacteria. It can be a virus, right? Virus infected cells. It can also be tumor cells, right? And additionally, when we start talking about things like transplants, this is part of the problem why we have to be very, very careful with transplants and having matching, finding that perfect match is to avoid immune responses such as natural killer cells. So a mismatched transplant would cause a huge reaction. Cells mutate all the time, right? And so when you hear things like killing tumor cells, it's maybe not as scary or as impressive as it might sound, because this is happening every single day, right? Natural killer cells release granules containing two particular types of enzymes that you need to know: perforin and granzymes. And the mode of action, or the mechanism that natural killer cells use to target these infected or tumor cells, is looking for the MHC1. So all of your white blood cells have MHC2, and all the other cells in your body have MHC1. And you can think of MHC as it's a receptor, and I call it the self-tag that says, I'm you, don't kill me. I'm part of the body. I'm a good cell, right? And so if the natural killer cell comes in contact with a cell that doesn't have the correct MHC1 or doesn't have any MHC at all, it's going to say, oh, you're an invader, or you're a naughty cell that something's gone wrong with, and we need to get rid of you. And then it's going to throw these granules full of perforin and granzymes and kill that cell. So perforin, it actually inserts itself into the plasma membrane, and that's going to cause an influx of water and then cause it to lyse open. And just to be certain that the cell dies, also have granzyme, which are protein digesting enzymes, and that digestion of the protein is going to trigger the cell to start a process called apoptosis. The cell very well might burst open from perforin before the granzyme actually does its job, but in case somehow the perforin doesn't kill, you have this backup system, right? So very potent way, natural killer cells are very good at killing these infected and tumor and mismatched cells. These are not, we don't consider this specific. It's any cell that doesn't have the MHC1, right? Okay. Now the granules kill the target cell. So when you're thinking about these infected cells, you're killing the target cell, but you're not necessarily killing the microbes that were inside the target cell. And so phagocytes will then have to come along and clear the actual microbes. But should be able to do that before the microbes can get inside another cell and infect it. Okay, so here's the differential white blood cell count. In a normal healthy individual, 60 to 70% of all of their white blood cells will be neutrophils. Only 0.5 to 1% of all of their white blood cells will be basophils. 2 to 4% of white blood cells will be eosinophils. 3 to 8 will be monocytes. And 20 to 25% of all white blood cells will be lymphocytes. Remember, lymphocytes include T and B cells. Most infections, most bacterial and viral infections, especially, will cause a drastic increase in the total number of white blood cells during an infection. But there are some infections that can actually lower your white blood cell count. And so we have terms to indicate when the patient is experiencing more or less. So leuco for leukocyte, white blood cell, right? Leukocytosis means more than usual. Leukopenia means less than usual. Now, leukopenia can also result from like chemo. Cancer treatments and other things can cause it as well. But if someone doesn't have any reason to have a drop in their white blood cells, then you would suspect that it is one of the types of pathogens that can lower white blood cells. And so that gives doctors, it narrows it down where to look. Okay, our patient isn't feeling well. We see lower white blood cell count. We know that they're not on any of these therapies that's known to lower it. So let's do some more tests and look specifically for these specific pathogens that are known to lower white blood cells, as well as maybe they would look for autoimmune disorder and other things that can cause it as well. But now they have areas to focus, right? So differential white blood cell count is very useful in finding the cause of illness or the cause of an abnormal differential white blood cell count, okay? The lymphatic system. Okay, so we just focused on the circulatory system in a way, right? We focused on the white blood cells. Now, the lymphatic system works hand in hand with the circulatory system. And you probably learned about the lymphatic system in anatomy, but I don't know how much you focused on the immunological aspects of it, so we will review that. Okay, so the lymph vessels found in all parts of the body except for your central nervous system. You don't find lymph tissues in your bone marrow either, and you wouldn't find it in tissues that don't have blood vessels, right? So wherever you have your circulatory system, you typically also have your lymphatic system. Fluid that leaves blood capillaries and is just sitting among the tissue is called interstitial fluid, as you probably remember. And this interstitial fluid, most of it goes back into capillaries of the circulatory system, but some of it remains and slowly drains into the lymph vessels, the lymph capillaries. And those capillaries, once the interstitial fluid enters the lymph vessels, we then refer to as lymph fluid. And the lymphatic vessels have one-way valves that shut to keep the fluid moving in one direction, similar to what you saw with the circulatory system in anatomy and physio. And this is really important. Our heart pumps blood through our body, right? But our lymphatic system is not connected to that. So our lymphatic system has some smooth muscle, involuntary muscle to help squeeze and move it, but a large amount of the circulation throughout the lymphatic system depends on our movement and our breathing. And I have a video that we're going to watch, and it's going to help reiterate all this information, but with really helpful visuals. Okay. You also have a few diagrams in your textbook that you can take a look at. Eventually, the lymph will go to either your right or left subclavian veins and return into the circulatory system as blood plasma. The lymphatic system has several functions like fluid balance, fat absorption, but of course, we are focused on the immunological defense function. And we'll see the most of that activity of providing support to the immune system will take place within lymph nodes. Lymph nodes have these special fibers that can trap microbes. They also have a high concentration of white blood cells, such as macrophages, dendritic cells, and your lymphocytes T and B. Okay, so remember, the macrophages and dendritic cells are going to be killing microbes by eating them up, phagocytosis, right? And then your T and B cells are going to work to provide adaptive immunity. And we'll talk about what they do next week when we talk about the adaptive immune system. So lymph nodes are testing centers, right? Scanning the lymph for the presence of microbes, and then killing microbes when they're found, and also initiating an adaptive immune response. Multiple large aggregations of lymph nodes or clusters of lymph nodes are found in the tonsils and also in the small intestine, called Peyer's patches. And this makes sense because as you breathe, drink, and eat, you're exposing your throat to all sorts of pathogens, right? So the tonsils are giving you a extra amount of protection at that what you can think of as like a weak point, right? A point of the body that's going to come into contact with a lot of pathogens. Similarly, in the small intestine, as you are undergoing a large amount of absorption of your nutrients into your bloodstream, you don't want any pathogens that have survived, hiding inside of food molecule, little, not molecules, but little food vesicles, I guess you could say. You don't want those to be absorbed, and you don't want them to start multiplying in the gut either. So Peyer's patches offer you a lot of protection there as well. So very strategic locations of these clusters of lymph nodes. And then, of course, you have lymph nodes throughout your entire body, as seen, you'll see in a second, and you'll see it in your book as well. The spleen is partly a lymphatic organ. It has other functions as well. It's not strictly lymphatic, but it is a giant. It's similar. You can think of the spleen as like a giant lymph node for the blood. Scanning the blood directly, it contains a lot of lymphocytes and macrophages. So in the same idea as lymph nodes. But lymph nodes don't have any other function. So lymph nodes are strictly looking through the lymph fluid for pathogens, right? And any debris and trash is also eaten by the macrophages. But the spleen has other jobs, which we won't talk about in this class. But it does also have the lymphocytes and macrophages to check the blood. So our body is very, very cautious, right? Is constantly checking the blood, either directly in the spleen or indirectly through the lymph. Okay. And then we have an organ called the thymus, which is also specifically dedicated to the lymphatic system. It is the location where T cells mature. That's why we call them T cells, after the thymus, site of T cell maturation. Okay. The thymus is interesting. It can shrink as you age, and for some people, it completely disappears. So I thought that was kind of a neat little factoid. All right, here is the video. This one is rather long for, usually I only show you 1 to 2 minute clips. This one I think is about 6 minutes, but it has such good visuals and a lot of really good information to explain the lymphatic system, which I feel the lymphatic system doesn't get as much attention as it really ought to, it's a major component of our immune system. Okay, let's watch.
The lymphatic system. Lymph vessels are found in all tissues except the central nervous system, the bone marrow, and tissues without blood vessels such as cartilage. The lymph system vessels are extensive as the vessels of the circulatory system. The lymphatic system serves several functions. It controls fluid balance by draining and cleansing the fluids that leave the circulatory system to deliver nutrients and gases to the tissues. It interacts with the villi in the digestive system to absorb and deliver fats to the circulatory system. It also has an immunological protection from viruses, bacteria, fungi, and cellular debris that could damage the cells of the body. From your understanding of the circulatory system, you know that the blood passes through the arteries, arterioles, and then the capillaries. The capillary walls allow the fluid portion of the blood to exit the capillaries into the surrounding tissues. Once the fluid leaves the capillaries, it is called interstitial fluid. About 90% of this fluid will diffuse back into the capillaries because of the difference in concentrations of the fluid. However, about 10% of the fluid will enter the open-ended lymph vessels. Once the fluid has entered the lymph vessels, it's now called lymph. These vessels eventually deliver the lymph to locations where the lymph can be cleansed of debris and checked for the presence of pathogenic organisms. How it gets the lymph there is pretty amazing. There is no heart for this system of vessels to pump the lymph around. So how does the lymph get to the locations it needs to be delivered to? The lymph moves through your body when you move your skeletal muscles. The contraction of skeletal muscles squeezes the nearby lymph vessels, pumping them. This pushes lymph through the vessels. In addition to the contraction of skeletal muscles, there are 2 other means by which lymph travels through the lymphatic system. There are smooth muscles at the larger lymph vessels. The contraction of these smooth muscles adds to the force provided by the skeletal muscles. Also, when we breathe, pressure changes occur in the thoracic region. When the thoracic pressure drops, that tends to pull lymph into the thoracic duct. One-way valves prevent the lymph from flowing backwards. The function of fluid balance is seen best perhaps when it goes awry. When the lymphatic system is prevented from doing its job, the fluids build up in the tissues. Edemas is the term given to this medical condition. Mild edema can occur during pregnancy when the weight of the baby slows the ability of the vessels to move the lymph up the body. More serious levels of edema can occur in a tropical disease called elephantitis, in which a parasite blocks the vessels, and the edema that is produced looks a lot like having legs of an elephant. Some lymph tissue is very diffuse with no clear boundaries. You can actually feel some when you rub your lower inner lip with your tongue. Others are more organized into groups, and these are called lymph nodes. Lymph nodes have 3 functions. First, they are testing stations. They monitor the blood by receiving samples of the blood plasma. Second, if the sample is rife with foreign invaders, they produce lymphocytes and send them into the bloodstream to try to destroy those invaders. In addition, the lymph nodes filter the lymph that they have so they can only return clean fluid back to the blood. Eventually, the lymph is returned to the circulatory system via the right and the left subclavian veins in the shoulders just above the heart level. Lymph nodules can be found as single structures in the body, or they can be grouped together in small clumps. That's what the tonsils are. They're groups of lymph nodules under the mucous membrane in the throat. These lymph nodules form a protective ring around the throat strategically located to protect the body from foreign invaders. If the tonsils get infected, they can become inflamed and abnormally enlarged, as you see here. This condition is called tonsillitis. If the condition is chronic, the tonsils can be removed in a tonsillectomy. Tonsils tend to get smaller as a person matures, and they can actually disappear altogether in an adult. Peyer's patches are very similar to tonsils. They're groups of lymphocytes and lymph nodules that are in the small intestines. Typically, they're found in the last third of the small intestine. Once again, they're strategically located to deal with foreign invaders. The lymphatic system's second function takes place here in the small intestine as well, the absorption of fats. We will discuss this more in depth in the topic of digestion, but
okay, so we're not really interested in the absorption of fats, so I'm going to skip to the spleen.
to the circulatory system. The spleen is a significant lymphatic structure and has a lot in common with the smaller nodes throughout the body. But unlike the lymph nodes, the spleen does not filter lymph. It's part of the lymphatic system, however, because it filters the blood. As the blood passes through the white pulp of the spleen, foreign invaders stimulate a response from the diffuse lymphatic tissue or the lymph nodules. The spleen also works to clean the blood of worn-out erythrocytes. Remember, red blood cells have a short lifespan. As a result, roughly 2 million erythrocytes die every second. They must be removed from the blood, and that's another job of the spleen. Before the blood leaves the spleen through the veins, it passes through the red pulp. Macrophages and the red pulp engage in phagocytosis to remove both foreign invaders and worn-out red blood cells. The third function of the spleen is to act as a reservoir for oxygen-rich blood. The spleen actually holds more blood than is necessary for its own metabolism. Therefore, it's an extra blood supply full of oxygen and nutrients. This serves as a backup supply of blood in case of blood loss. If the body detects blood loss due to hemorrhage, the sympathetic division of the ANS stimulates the smooth muscles in the capsule of the spleen to contract. This pushes the backup supply of blood into the bloodstream, compensating for the blood loss. Although the backup supply of blood in the human spleen is rather minor, it's a major factor in the physiology of some other mammals. Seals use the spleen as a built-in oxygen tank. When the seal dives, it conserves its oxygen as much as possible. However, when it's running low and cannot get to the surface, the smooth muscles of the spleen contract, sending the oxygen-rich blood stored there into the bloodstream.
So as interesting as this additional function of the spleen is, for our class, for immunity, we're only concerned about the presence of the T, B cells and macrophages to look for foreign invaders in the blood.
This gives the seal more time before it must surface to breathe. Although the spleen is part of the lymphatic system, you can live without it. If your spleen is ruptured due to an injury, it can be removed in a splenectomy. This is often necessary in order to stop internal bleeding because the spleen is so vascular. Once your
Okay, so let's skip to the
like
thymus.
the tonsils, the thymus gland changes as a person matures. When a person is young, the thymus gland is large in proportion to the body size. During this stage of life, it is mostly lymphatic tissue. After puberty, it decreases in size and becomes mostly fibrous and fatty tissue. What does the thymus gland do? Like many things in the human body, the scientific community is still rather puzzled by the thymus gland. We know that while a person is young, immature lymphocytes known as T lymphocytes leave the bone marrow. Remember, blood cells are made in the bone marrow, and they travel to the thymus. Through a remarkable maturation process, sometimes referred to as thymic education, T lymphocytes that are beneficial to the immune system are spared, while T lymphocytes that might evoke a detrimental immunological response are eliminated. For example, if you have type A blood, T lymphocytes which attack the A antigen are destroyed. However, T lymphocytes which attack the B antigen are allowed to mature and enter the bloodstream. Notice that this one is called a gland. That means that one of its functions is to secrete hormones, making it also a part of the endocrine system as well as the lymphatic system. It produces hormones. Principal among them is the hormone thymosin. What does thymosin in the body do? Well, we're not really sure. We know that it affects the immunological response of the body. However, the way that it's done remains unclear. One prevalent thought is that thymosin stimulates the activity of lymphocytes to migrate to other lymphatic tissues.
Okay, so that is the lymphatic system. Now we'll focus on phagocytosis. So in Greek, phago means to eat and cyte means cell. So phagocytosis are cells that eat and they are specialized white blood cells that ingest microbes, cell debris, right, particles. So you can think of them as the vacuum cleaners of the body. And of course, we're focused on them for their their ability to take in microbes and kill them. So only some of the white blood cells are phagocytic, and that would be your neutrophils, your macrophages, and your dendritic cells. And again, remember, macrophage and dendritic cells come from monocytes. And here's an image showing the macrophage, which is this large yellow bumpy cell that you see, and some bacteria shown in red. So these are some rod-shaped bacteria. And the macrophage senses that they're present and extends these pseudopods, wraps around it, and engulfs it and brings it inside and kills it. So let's see what those steps are. Before we look at the steps of phagocytes, remember that both granulocytes and monocytes have to leave the blood and migrate into the tissue to the site of infection. And that's especially important when we're thinking about the monocytes, right? Monocytes, they will enlarge and mature into macrophages or dendritic cells once they're in the tissue. The initial stage of infection, you'll see a very large increase in the number of neutrophils, which will take care of most of the infection, but they will be, eventually macrophages will take over this job. And so then the number of neutrophils will decrease back to near normal, and the amount of macrophages will be very high. Okay, and you would be able to detect that in the differential white blood cell count. Okay, so the steps of phagocytosis. Chemotaxis, the being drawn towards a chemical stimulant, and in that case, it could be things that are being released from the microbe, but it could also be chemicals that are being released by other cells to alert the phagocytes that there is a problem. And then adherence, the extension of those pseudopods, right? So step one, chemotaxis and adherence to the phagocyte. And in this image, they show one way in which phagocytes can grab on to bacteria is through this toll-like receptor and a part of the pathogen called PAMP. Remember, we learned PAMP stands for pathogen-associated molecular patterns, and that can include things like flagella or peptidoglycan, other parts of microbes. And so this is showing one way that you can get adherence to through those extensions of those pseudopods. Okay, then it's going to, the phagocyte's going to bring the microbe inside of itself, so ingest and form a vesicle. And the vesicle is called phagosome. And then the lysosome vesicles inside of the phagocyte is going to fuse with the phagosome. And so you see it here, the plasma membrane of the lysosome fuses with the plasma membrane of the phagosome. And then that creates what we call a phagolysosome. The lysosome contains lots of digestive enzymes and toxic oxygen that will kill and break down the microbe. And so that's what we refer to as digestion. And then that's going to lead to the little bits and pieces that we're not able to digest, which will then just be waste that gets eliminated from the white blood cell that will eventually be, it'll be either part of it'll be immediately part of your interstitial fluid, and it will either get taken up by the capillaries of the circulatory system or the capillaries of the lymphatic system, and then it will be excreted from the body in your urine. So yeah, that's the cycle of phagocytosis. Okay, so here's a very short video just to show you those steps in action, but it's an animation. we don't want their little intro.
When bacteria or other pathogens are present in the body, certain white blood cells, categorized as phagocytes, consume the microorganisms to protect the body from infection. The process of consuming the pathogen begins when the phagocyte is attracted to chemicals from the pathogen and, through receptors, binds to it. The phagocyte ingests the microbe, encasing it in a compartment called a vacuole. Lysosomes in the phagocyte merge with the vacuole. Its enzymes then kill and digest the pathogen and release any indigestible remains from the cell. This cell process, called phagocytosis, protects the body by removing pathogens.
All right. I think that was the only video I had for phagocytosis, right? Yes. Okay. So a little bit more detail about those lysosomes that fuse and lead to digestion and killing of the microbe. So you have lysozyme, which we talked about earlier in part 1, hydrolyzes or breaks the bonds between NAG and NAM to break down peptidoglycan. We also have lipases to break down fats, proteases to break down protein, ribonuclease and deoxyribonuclease to break down nucleic acid, right, RNA and DNA. And so this is the digestion part of the lysosome. And the digestion doesn't take place within the lysosome. It takes place once the lysosome fuses to the phagosome and you have the phagolysosome, right? Okay. And then in addition, there's a bunch of other enzymes within the lysosome that will create toxic oxygen species. Things like superoxide and hydroxyl radicals. We have hydrogen peroxide, we have nitric acid or oxide. And so all of these different toxic reactive oxygen species to kill the microbe. And this is important because we don't want the microbe to start multiplying and dividing inside of our white blood cell. However, you'll see that there are some microbes that have figured out ways to bypass phagocytosis. So we'll see that. So just a little review of phagocytic cells and their location in the body. Neutrophils are very numerous, the most numerous. They circulate in the blood all the time, and they're also throughout our tissues. Monocytes are inactive, right? Monocytes are the inactive form. They circulate in the blood. They will be attracted to the site of infection, in which they will change into macrophages or dendritic cells. Okay. Tissue macrophages, these are macrophages that stay in their particular tissue. You can think of these as like guards of the particular tissue. We have broken those into different groups, some called histiocytes, which are in the spleen, lymph nodes, and bone marrow. And then we have some that are only found in the liver, and those are called Kupffer cells after the person that discovered them. Then we have alveolar macrophages, which are only found in the alveoli of the lungs. And then we have the microglia, which are super specialized to function within the central nervous system. And as I mentioned, and as we've seen this pattern throughout the entire class, is that microbes will evolve to overcome barriers, right? So there will always be these exceptions, just like we saw with resistance to antimicrobial drugs. We see these evasion strategies in different microbes, but we don't call it resistance, and we don't see this like spreading of these traits. It seems to just be very specific types of microbes that have these natural properties that evade phagocytosis. Okay, and then we'll see that again when we look at the complement system. Okay, so Streptococcus pyogenes, remember the causative agent of strep throat. It has these special proteins called M proteins. It also is can make capsules, and this will make the phagocyte, such as a neutrophil or a macrophage, is not able to grab onto it, right? So Streptococcus pyogenes has the M protein, whereas we see pneumoniae has the capsule, and both of these make it hard or even impossible for the phagocyte to grab onto it. And so they will not be taken up by phagocytes. Then we have Staphylococcus aureus, which we talked a lot about when we've talked about resistant bacteria, right? Like MRSA, for example. And Staph aureus is able to just straight up kill the phagocyte with a special chemical or toxin that it releases called leukocidin. Okay, we have Listeria that is able to produce its own type of membrane attack complex that will lyse open the phagocyte, which is super interesting because we have a system, you'll see at the end of this lecture, where we actually make a membrane attack complex to fight off bacteria. And so now this Listeria actually does the same thing to our phagocytes. So that's very fascinating. Then we have Shigella and Rickettsia. And remember, Rickettsia, Rocky Mountain spotted fever, for example, all of the spotted fevers are different Rickettsia bacteria. They're able to escape the phagosome, and so is Shigella. And we don't have the mechanism. We're not going to look into detail how they do that, but that's very interesting in that they can be taken up by phagocytes and the phagosome will form, and then they will escape, and the rickettsia will actually multiply within the phagocyte. It'll just start, because rickettsia is an intracellular pathogen. So it's like, great, now I'm inside. Hooray for me. And then we have things like Mycobacterium tuberculosis and HIV virus, right? And they can prevent the lysosome from fusing to the phagosome so it doesn't get digested. And it can just survive. And then another non-specific survival, we have Burnetti is able to actually survive in the phagolysosome, which is incredible. It must have a lot of very complex mechanisms that allow it to inactivate all those enzymes and all the toxic oxygen. And so that's the evasion of phagocytosis. Continuing with the second line of defense. So we've talked about immune cells of the innate immune system, phagocytes. We've talked about natural killer cells. Now we're going to shift gears into inflammation, and then we'll talk about fever, and then we'll wrap up the whole chapter talking about briefly antimicrobial substances. Okay, so inflammation is a local defensive response. It's caused by tissue damage. Tissue damage can be caused from microbial infection. It can be caused from physical agents like too much heat, you burn yourself, right? Radiation, electricity, or you cut yourself on something, right? A sharp. Chemical agents, gases, acids, bases, right? All of these can cause tissue damage and would cause local response to that damage called inflammation. Inflammation, the functions of inflammation is to, if there's a microbe, kill it, keep it from spreading. Whatever is causing the tissue damage, we want to wall it off, keep it from spreading throughout the body, right? And remove it if possible. And then we want to repair the tissue that's been damaged. Some tissue damage will result from the process of dealing with the microbe or injurious agent, right? So the initial tissue damage is from what we saw above, right? These different ways to damage tissue. So that causes the initial tissue damage, but turns out steps 1 and 2 can cause some tissue damage as well. And so tissue repair starts immediately to repair the damaged tissue and will continue on until our inflammation is completely finished and all of the tissue is completely repaired and replaced. And so symptoms of inflammation, pain, redness. If the inflammation is severe enough, you might have a local loss of function. An example of that is if you, if you've ever had an injury to, for example, like your knee, right? And it becomes so inflamed that you can hardly bend it anymore. That would be an example. But the loss of function is not just a movement, right? It could be if it's in the liver and your liver becomes severely inflamed, you may lose temporarily the ability to properly, filter the blood, which the liver does, and the liver does a whole lot of other functions as well. You could have jaundice as a sign that the liver is not functioning. So inflammation is a local loss of function. If the inflammation is severe enough, whatever that tissue is supposed to do normally might not be able to do it while it has that severe inflammation. Swelling is due to the accumulation of fluid. So you have this huge influx of blood flow, and now you have also this huge activity of immune cells. And so all of this is going to cause fluid. And so you can have swelling. And so sometimes immobility can also result from the swelling, which is kind of the example I gave with not being able to bend your knee or your finger if you sprain something. And then a lot of times with inflammation, you'll have some extra heat, which is the extra blood flow, right? So pain, redness, possibly immobility, but not always, possible swelling, but not always, and heat might not be detectable to you, but will be present. Okay, we typically will have acute inflammation, which is, when you have this injury, you'll have acute inflammation as a rapid, quick response initiated by neutrophils, but then, other white blood cells will join in. It develops very rapidly and it usually lasts a few days to a few weeks. You experience acute inflammation every time you have a cold or flu or any other, temporary illness or injury. Chronic inflammation can develop more slowly, it can be quite severe, can last months to years. And because it's so long in duration, it's mostly caused by monocytes. Remember that turn into macrophages. So it's mainly caused by macrophages instead of the neutrophils of the acute inflammation. An example of an infectious disease that can cause chronic inflammation would be tuberculosis. So how does this whole process get started? So I mentioned tissue damage, and that's true. And when cells are damaged, they'll release cytokines. We also have, we have macrophages and neutrophils that are constantly checking the area for invaders. And if they find an invader. They would know that they found an invader when the invader's PAMPs trigger the macrophage or white blood cells TLRs. Okay, so if a TLR of a macrophage gets triggered, it's going to release cytokines, and this is going to cause two things to happen. Okay, the cytokines are going to activate acute phase proteins that are already in circulation in the blood. It's also going to tell the liver to make more acute phase proteins. Okay, and so you were probably asking yourself, well, what the heck is an acute phase protein? Good question. So some examples of acute phase proteins are the complement system's C-reactive protein, which we'll learn more about this in a few minutes. There's a mannose binding lectin that's always in circulation to detect mannose. Mannose is found on many bacteria. We also have kinins, which is a type of, I would say it's a type of cytokine for vasodilation, but it's an acute phase protein. So yes, it's a messenger to cause vasodilation, but it's doing that under this acute response, right? So it's an acute phase protein as well. And then there's other specialized proteins for clotting, right? Because if we have tissue damage, we need to take care of that right away. So there's all sorts of acute phase proteins in the body. Vasodilation, the purpose of that is to bring in more blood, okay? Then you also need to increase the permeability of the vessels themselves so that as you bring in more blood, those white blood cells are able to squeeze through the blood vessel and get into the tissue. So the first stage of inflammation is that vasodilation and increased permeability. So immediately upon damage, cells release histamine. This especially is true for those mast cells that we talked about. But any cell that gets damaged is going to release histamine. It's going to dilate your blood vessels, open them up, make them bigger so that you get more blood flow, essentially so you can get more of your white blood cells to the area of damage. You're also going to get an increase in permeability from the presence of histamine, and that helps the phagocytes to leave the blood and enter the damaged tissue. It also will, as a side effect, allow more fluid to leave the capillaries, which can lead to the swelling and more pain as well. Kinins, their job is also to promote vasodilation and permeability, but they also have another function of being very attractive. So this will promote chemotaxis to attract neutrophils to the injured area. Prostaglandins are also released by damaged cells to intensify the activity of kinin and histamine. So an amplification of blood vessel dilation and permeability. They're also going to help phagocytes to move through the capillary wall to get from blood into the tissue. And we do also associate prostaglandins with an increase in pain. Prostaglandins tend to get a bad rep for causing pain, but they're actually super important for this protective response of inflammation. Leukotrienes are going to be a special cytokine released by mast cells and basophils that help phagocytes to do their job. So they enhance phagocytosis by helping phagocytes attach to the pathogen. Also, during this first stage of inflammation, blood clots form, right? Stop any blood loss that's happening. This also will help wall off any microbe or toxins that are present so they don't spread throughout the body. But this clot may result in an abscess sometimes. an abscess is a localized collection of pus. Pus is a mixture of dead cells and body fluids. Pus can then collect in a cavity. This cavity would be formed by the breakdown of body tissue. And so this is where you see that the immune response can cause damage. So you have some initial damage in the body that causes this response, but then you do get some additional damage as all these white blood cells are doing their job. And so that can lead to a cavity and a collection of pus, resulting in an abscess. Common names or examples, types of abscesses can be pustules or boils. So this is showing that first step of the tissue damage happening. And then this is showing the actual first stage of inflammation where the blood clot forms. You have the release of all of these messengers to bring in extra blood flow and allow white blood cells to start to come to the scene. And that's where you can also have the abscess form. So this is a summary, everything we just talked about now in a nice chart for you, so you can memorize the specific functions of histamine, kinin, prostaglandins, leukotrienes, cytokines, additional cytokines, and the complement system. And you'll see that certain cells will release certain messengers or mediators of inflammation. Okay. So yes, you need to memorize this chart. Okay. The second stage of inflammation is phagocyte migration and phagocytosis. So really my title is incomplete. It should say stage 2 of inflammation is phagocyte migration and phagocytosis. I just ran out of space. Margination, diapedesis, and then phagocytosis occur during this second stage of inflammation. Margination is the specific mechanisms that cause phagocytes to stick to the inner surface of the vessel so that it can get ready for leaving the vessel. Cytokines cause alteration of the lining of the vessel to have a adhesion molecule, so the adhesion molecules on the endothelium or the inside lining of the blood vessel, and this grabs on to the phagocytes as they are passing through the blood. And there's two stages. There's a slow rolling from like a weak interaction, and then there's a completely stopping of the white blood cell, so a very strong bond, and then that really leads into allowing the white blood cell to squeeze through. So diapedesis, those phagocytes that have been stopped and adhered to the lining of the blood vessel then will squeeze between the endothelial cells. Remember, blood vessels are a single cell thick, right? So it only has to squeeze between two cells and then it's out, right? Capillaries are single cell thick, okay? And so then they squeeze through the capillary cells and they then are in the tissue, and they will continue to migrate through to the area of the damaged tissue. And then they can begin their phagocytosis, which we already studied the steps of phagocytosis. So here we remind you that in this initial stage, most of these phagocytes will be neutrophils. The majority will be neutrophils. They will begin to ingest and destroy the microorganisms. Eventually, macrophages will take over this job. And so this summarizes the second stage of inflammation, which has margination, diapedesis, and phagocytosis. This image is from your textbook. here is an animation of the process of the white blood cells adhering to and squeezing through the vessel.
Extravasation of neutrophils. White blood cells circulate in the blood, searching for pathogens within our bodies. Neutrophils, the most abundant type of white blood cell, patrol the blood for signs of infection. When inflammation is detected, neutrophils become sticky and roll along the capillary wall, adhering to it, and eventually squeezing out of the vessel. This entire process is called extravasation. Extravasation begins when chemokines are released by immune cells at a site of injury or inflammation, activating the endothelial cells of nearby capillaries to express adhesion molecules. Neutrophils respond to the adhesion molecules by expressing counter receptors, which produce a weak interaction between the neutrophil and endothelial wall. The neutrophil adheres slightly to the vessel wall, rolling along the surface. This movement is possible through unique molecular interactions between neutrophils and the endothelial cells. The neutrophils are slowed down by the formation of weak transient bonds between Lewis X, a carbohydrate expressed by neutrophils, and selectins, adhesion molecules produced by endothelial cells. Searching for pathogens.
Shoot, I didn't mean to rewind. I just wanted to pause. So as this video tells you about the specific mechanism of the white blood cell to adhere to the lining, I'm not going to test you on all the names of the molecules.
Adhering to it, and eventually squeezing out of the vessel. This entire process is called extravasation.
So we are calling it diapedesis, specifically the squeezing through the two cells of the capillary is the diapedesis, and the getting to that location, we are calling it margination. So they're lumping both processes together. We are specifying that there are two different processes, right? First, getting attracted, having those cytokines cause this physiological change in the vessel so that the binding can occur, and then the actual squeezing through. Okay.
Extravasation begins when chemokines are released by immune cells at a site of injury or inflammation, activating the endothelial cells of nearby capillaries to express adhesion molecules. Neutrophils respond to the adhesion molecules by expressing counter receptors, which produce a weak interaction between the neutrophil and endothelial wall.
Okay, again, so we're calling that margination.
The neutrophil adheres slightly to the vessel wall, rolling along the surface. This movement is possible through unique molecular interactions between neutrophils and the endothelial cells. The neutrophils are slowed down by the formation of weak transient bonds between Lewis X, a carbohydrate expressed by neutrophils, and selectins, adhesion molecules produced by endothelial cells. The force of blood flow causes neutrophils to roll along the endothelium, causing new interactions to form while old ones break. Eventually, tight binding occurs when integrins known as LFA1 on the neutrophils undergo a conformational change, causing them to bind firmly with endothelial adhesion molecules called ICAM1. With the neutrophil firmly adhered, diapedesis, the process of neutrophils exiting between endothelial cells, can begin. Once outside the blood vessel, the neutrophil migrates to the infection, guided by chemical signals.
Okay. So after the phagocytes have their margination, diapedesis, and they perform all their phagocytosis, and the issue is resolved. All microbes are gone, dead, all the whatever damaging agent might have been present has been taken care of, then tissue repair can finish. But again, remember, tissue repair starts immediately during stage 1, but it's not able to be completed until all the harmful substances or microbes have been removed and the immune response itself is diminishing. The stroma and the parenchyma produce new cells to replace the damaged cells. Now, if this replacement or production of new cells comes from the parenchyma, then you have a great reconstruction that has all of the function and it'll look the same. And so that's when you don't really get a scar, right? It'll be perfect resemblance because it'll be made of the same exact cells. If the damage is, severe enough, and the stroma do the majority of the replacement, then that's when you get the formation of scar tissue. So you'll notice paper cuts don't ever lead to scars, right? Because it's not going to be deep enough for the stroma to be involved, right? But a deeper, very deep cut, such as something that requires stitches, typically will leave some amount of scar. So when the stroma is responsible for the replacement of the tissue, that's when you get the scar. And of course, my examples are just thinking of the skin because that's what you can see. But of course, this can be taking place in any area of the body. And scar tissue can be really problematic when we're talking about organs because they are not made up of specialized cells and they therefore don't have a function. They are not able to do the job that tissue is supposed to do. And so if you have a large amount of scar tissue in the liver, for example, you can have permanent compromised function of the liver at that point. If it becomes severe enough, we call it cirrhosis. Okay. Skin has a high capacity for regeneration. So our skin is really good at healing because it's coming in contact with everything all the time, right? It needs to be able to sustain injuries and heal. But our cardiac muscle, for example, really should never be damaged. And so it really doesn't have a capacity to regenerate. It has a very low capacity to regenerate. So whenever damage to the heart occurs, it's a very concerning. It's not good, right? So this image shows you this, the scab has formed and will start to diminish as it gets replaced with the tissue. Same thing with the blood clot underneath it. This will get smaller and smaller as the regeneration of those cells occurs. And this is saying what I had said already out loud, but remember that the stroma is the supporting or connective tissue, right? And the parenchyma is the actual functioning part of the organ. And so we see, the perfect replacement with its normal function when the parenchyma is are the cells that are replacing the damaged cells. Chronic inflammation tends to cause the stroma to be active, leading to the buildup of scar tissue called fibrosis. Fibrosis typically interferes with the function of the tissue as those scars are not specialized cells, right? And this is especially common in microbes that evade phagocytosis, and that stage 2 just continues to persist and is taken over by macrophages. So chronic inflammation, which is attributed to by macrophage, is going or has the potential to build up scar tissue and be very problematic. All right, fever is a systemic response, right? It affects the entire body. And we tend to, in society, we don't like fevers, right? Because you don't feel good, you're ill. But fever is actually very beneficial as long as we keep an eye on it and don't allow it to get out of control. How does it happen? When your hypothalamus, which controls your body temperature, is told to reset its set point to a higher temperature. How does that happen? Certain cytokines tell the hypothalamus to release prostaglandins, and then those prostaglandins reset the hypothalamus to a higher temperature. So then suddenly your body receives this message that it's supposed to be, 39 degrees instead of 37. And now it thinks, well, we are way too cold. We need to warm up. So vasoconstriction will happen. So bringing all the blood closer to the inside of the body and less blood flow to your extremities. This is to keep it warmer, right? Decrease the amount of heat that's lost. Shivering, right? Shaking of your muscles, rapid shaking of your muscles is going to produce heat. Also, speeding up the rate of metabolism is going to produce more heat. And so that's how we get more heat in our body. And that fever will continue as long as those cytokines are being released and are present in the hypothalamus. Then prostaglandins will continue to be released, and the fever will continue. So when you take things like Tylenol, a fever reducer, it's blocking those prostaglandins so that the fever doesn't occur, or it doesn't occur as dramatically. But I would recommend that you don't do that unless told to by a physician, because fever can slow down or even kill a pathogen. It also helps your immune system to speed up and work faster. So you'll get an increase in the proliferation of and the activity of T and B cells. You'll have an increased rate of tissue repair, and you'll also have an increased rate of interferon production. We haven't talked about interferon. We will in a few minutes. Disadvantages, of course, if the fever gets too high, you can have delirium, brain damage, death. It does speed up metabolism and all these processes. So that can speed up your heart rate. And for some individuals, they're more sensitive. They might have underlying health conditions, and so it can be an issue. For most normal individuals, a fever would not be an issue, right? Dehydration can occur for anyone, though. Now we have more activity happening, we're hotter, so we need to replace our fluids more rapidly. So I included this when is a fever harmful, just for your own information. I'm not going to test you on these because these are in Fahrenheit, because when we take our temperatures, we're typically looking at Fahrenheit, right? Because the United States doesn't follow the metric system, annoyingly. So just to be useful and helpful, I just did it as Fahrenheit. Okay. So babies 3 months and younger, they're very sensitive, and they should not ever have a fever above 100.4. So if you have a baby 3 months or less and their fever starts to get to 100.4, you should immediately call the doctor and find out what they advise. Okay. 3 months up through adolescence, they can sustain a fever of 100 and up to 102. If it becomes 102, you should call the doctor. Adults, we can handle a little more, 103. If your fever is 103 or more, you should contact your physician and see what they advise you to do. Again, you don't have to memorize this information here. It's just to help you. When the hypothalamus is reset to 37 degrees Celsius, your body's suddenly going to be told that it's too hot, right? And so now you're going to send lots of blood to the surface of your skin to cool it off. You're going to sweat to increase the coolness of your skin, right? As sweat evaporates, it takes a lot of heat away from the body. And so this is when you know your fever has ended, has stopped. It doesn't mean that you won't get it again. Sometimes as you have an illness, you'll have a fever and it breaks and then it comes back, and it does that for a couple days. Other times you just have it consistently until it's done. Okay. When the hypothalamus is reset, though, it will cause that that rapid cool down, which we call crisis. And that's a little misleading because it sounds like it's dangerous and bad, but it's actually, good when you're better and you're getting back to a normal temperature. Okay. So antimicrobial substances, we will be talking about the complement system, interferon, and then something called antimicrobial peptides, and that will be the end of our lecture. So the complement system is not really part of the innate or adaptive. It's kind of it interacts with both, but because it's always present and it doesn't necessarily react to specific pathogens, we are including it as part of the innate immune system. Okay, what is it? It's a series of reactions of proteins that lead to increased inflammation, increase phagocytosis, and increase direct killing, so cytolysis. Okay, we have three main pathways that it can be activated: classical, alternative, and lectin pathways. All three of these pathways, though they are initiated in a different mechanism, eventually get to the same step of splitting the protein called C3, and that is what will lead to the effects of the complement system. So I want you to realize that we have different complement proteins that are always in circulation in the blood, and when they get activated by the presence of a pathogen, then you will get the actual outcomes of the complement system. Okay, so the classical pathway, and it's called classical only because it was discovered first. This is when you actually have antibodies that have attached to a microbe. So this is where you can see the adaptive immune system has played a role, because you already have these antibodies that are attaching to the microbe surface. And then that the ends of the antibodies that stick out can bind the C1 complement protein, and that causes it to break in half or cleave. And that begins this cascade of all these proteins splitting, and then you get the outcome eventually. And we'll look at those outcomes in more detail in a little bit. We'll also see images of these pathways and a short video. For the alternative pathway, instead of it being an antibody triggering the C1 to split, you now have different factors that we call B, D, and P, which are always present in the blood. They bind to the antigen, and they cause a different complement protein called C3 to cleave and start that cascade that leads to the outcomes of complement. And then lastly, you have the lectin pathway, where that mannose binding lectin that you always have circulating binds to the antigen and activates a different complement proteins called C2 and C4. All right, so let's look at that. So this is the classical pathway, which involves the adaptive immune system, because now we have antibodies. When you have two antibodies side by side, they can bind the C1 complement protein, which is always circulating in the body, and lead to the production of C2 and C4. And those will then split, and eventually those products will cause the C3 to split. And it's when C3 is split is when you get increased phagocytosis, you get the formation of a special system that will cause direct killing, and you get an increase in inflammation. We'll look at these in a little bit more detail in a minute. Okay, so I do not want you to memorize C2 splits into C2b and C2a. I don't want you to memorize C4, C4a, C4b, but I do want you to memorize, right, that the microbe is coated in these antibodies. The two adjacent antibodies trap the C1 complement, C1 protein, starting the cascade, which leads to the splitting of C3. Okay, and it's when C3 splits that you get the three outcomes of the complement system. Okay. The alternative pathway, instead of antibodies, you have these factors in the blood that are always circulating in the blood called BDP, and they can coat the surface of the microbe and directly split C3. And so this is shorter, much easier pathway. So you do need to know everything here, right? So some of the receptors on microbes are these lipid carbohydrate patterns that will bind to these factors in the blood called BDP, and those are going to directly cleave the C3 protein. C3 is also always in the blood. And then the cleavage of C3, just like you saw here, the cleavage of C3 will always lead to opsonization, cytolysis, and inflammation. What is opsonization? Opsonization is when this C3b protein actually coats the surface of the microbe. So a bunch of this gets made and coats the microbe. And that makes it much easier for phagocytes to grab onto it. So it'll actually attract and help the phagocyte to bind to that microbe and eat it up. It also starts a cascade that leads to the formation of what we call the MAC, membrane attack complex. And then the other protein, the C3a, will lead to increased inflammation in the area to fight the infection. Okay, then you have the lectin pathway, which instead of having BDP bind to receptors on the microbe, you have this lectin that binds to mannose. So you have other receptors on a microbe that are made of mannose, and you'll get the binding of lectin to the mannose. And so your book calls this mannose binding lectin to help you remember that it's lectin is binding to the mannose, which is a type of sugar carbohydrate. So a different receptor commonly found on microbes. That leads to a whole cascade. Okay, the initial step that you need to know is that the lectin will activate both C2 and C4. That will lead to the activation of C3. So you don't need to know this intermediate step. And then C3, as we've already talked about, leads to opsonization, cytolysis, and inflammation. Okay, so what are these outcomes of complement that I keep talking about? Right, inflammation, is the increased blood flow and permeability. Also will help attract phagocytes to the area. Opsonization is an enhanced function of phagocytes to be able to grab onto the pathogen. So the pathogen will be coated with C3b, and then the phagocyte can grab onto the pathogen much easier and more quickly. And then cytolysis, as I mentioned, is a whole bunch of different proteins, and you do not need to memorize the numbers here. But once you have the cleavage of C3, this C3a is going to go split into a bunch more proteins and embed into the plasma membrane of the bacterial pathogen. And then that will cause a small hole, which will allow the influx of water into the cell and burst it open. So here is an image. Here's the bacteria right before and right after cytolysis. All right, here's a brief video on the complement system.
The complement system is an essential and powerful part of our immune defense system. Its main function is to coordinate the destruction and removal of infectious organisms, known as pathogens, or damaged cells in our body that need to be replaced. The complement system consists of a cascade of proteins, which activate each other by cleavage, the process of splitting each protein one by one. The complement system may be activated by 3 different pathways: the alternative, classical, and lectin pathways that all run through complement C3, the central protein in the cascade. C3 serves as the master switch, controlling all downstream effectors that ultimately cause destruction of the cell. The alternative pathway is spontaneously activated through direct binding of complement to a pathogen or damaged host cell to help the body clear away debris. The classical pathway is activated when an antibody, a protein produced by our immune system, recognizes and binds to a component on the surface of a foreign cell, forming an immune complex, starting an immune response that targets the pathogen for destruction. The lectin pathway is activated by natural molecules in our body that recognize sugars at the surface of infectious organisms. In situations where the complement system becomes overactivated, it results in immune attack, the destruction of healthy cells, and ultimately causes illness to the person.
So I didn't mention this, but the complement system can be overactivated sometimes, which, because it's such a powerful amplification of inflammation and phagocytosis, that can lead to tissue damage. And so we actually have mechanisms in place to try to prevent that from happening. But, there are times that it still can happen. So just like we saw with phagocytosis, there are some bacteria that have mechanisms to evade complement. And I'm not giving you any specific bacteria for this, but I will tell you in general that bacterial capsules can prevent the complement protein from being activated. Bacterial surface complexes, so certain bacteria have these special surface proteins that will actually prevent the membrane attack complex from forming, so that C3A is not able to, it will split into all those additional proteins, but those little proteins will not be able to embed into the plasma membrane of the bacteria, only for the bacteria that have those specialized complexes to prevent it. And then lastly, some bacteria will secrete enzymes that will digest different complement proteins. And so this might, they might have enzymes that only block out one of the functions of complement, whereas others might even block out all. It depends on which which complement protein is being digested. So I cannot give you really any too specific definition here, but so what you have to remember is that it can prevent any or even all of the functions of the complement system, depending on which complement protein gets digested. It's most commonly we see that most of the enzymes caused by these specialized bacteria. So only some bacteria can evade complement, and only some of those have these enzymes to digest complement. And the most common one that we see is that the inflammation is either reduced or shut off. And so remember that C3A really is responsible for that inflammatory response or outcome of complement. And so it'd be the most common enzymatic digestion is of C3A. Okay, so now we're going to look at interferons. I think we've briefly talked about interferons already, but let's review and expand. Also, this is a very brief overview of interferons, but interferons alpha and beta are specifically helpful for viral infections, and that's what we'll look at an image of next, is that once a cell becomes infected, it will start to secrete interferon alpha and beta so that its neighbors know that the virus is coming, and they can start making antiviral proteins and enzymes so that they don't become infected. Interferons are messengers that cause a reaction, right? So these are a special category of cytokines. Interferon gamma, on the other hand, is when neutrophils and macrophages need to phagocytize bacteria. So interferon gamma will cause neutrophils and macrophages to increase their phagocytosis, right? Whereas interferon alpha and beta are specifically for antiviral function. And so here, this is looking at a cell that has already been infected and released interferon. And this cell that you see here is the neighboring host cell. So a neighboring cell receives the interferon signal that says, watch out, a virus is coming. And as you can see, the virus is coming. But before the virus is able to completely replicate, the interferon has already caused the cell to produce these antiviral proteins that stop the replication of the virus. And so now this virus, this cell has been spared from true viral infection, because this virus that entered was not able to replicate. And so this is very important mechanism for fighting viral infections. And this is therapies with interferons are becoming more and more popular, because they are telling the cells to make very specific products to fight and stop infection. Turns out the body already has a lot of antimicrobial peptides to fight infection. We're learning about them. So I don't have a whole lot to tell you yet about these antimicrobial peptides, but what we do know, they're short, they're peptides, right? Only 12 up to 50 amino acids in sequence, so they're short peptides. And there's all sorts of different types that have different functions, and they get produced when TLRs are triggered. So another function of TLRs being triggered, we see like sweat glands, neutrophils, macrophages, epithelium, and even platelets can produce AMPs. Some AMPs kill microbes directly. Some of them will do this by cell wall synthesis inhibition. Some of them will form holes in the plasma membrane, and some of them will destroy DNA and RNA of the microbe. So killing the microbe by these 1, 2, 3 mechanisms. This is the main way that the AMPs are working. But we also see another important function of AMPs is to sequester or like group together, get rid of LPS. So LPS, lipopolysaccharide, we talked about, when there's enough of it, it can cause an overstimulation of the immune system, which would lead to shock. So we call that endotoxic shock. So AMPs are really good at helping to reduce the likelihood of endotoxic shock. They also attract dendritic cells to increase this phagocytosis and initiate adaptive immunity. And they can also attract mast cells to increase blood vessel permeability and vasodilation. So they play a very important role in all sorts of areas of the immune system, right? So very interesting, new kind of new field of study. And that is it. I have included a study guide this week. So in addition to these practice questions throughout the lecture, at the end of your chapter, you have more practice questions, and then you have study guide. And at the end of the chapter of the study guide, I included exam questions, a few exam questions for you to practice and see the types of questions that I'll be asking. All right, that's it. Good job. Have a good day.